Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and in this module we were discussing about the affinity chromatography, and in the previous lecture we discussed about the basic principle of affinity chromatography, what are the determinants are playing crucial role by bringing the receptor and the ligand together. And we have also briefly discussed about the different types of matrix what you can use. So, in a typical matrix what you have is you have the coupling of the receptor or the ligand to the matrix, and then you can use the couple the cognate pair. For example, in the case of matrix you can couple the ligand, or in the case of ligand you can couple the matrix to the protein of your interest, and then subsequently you can be able to utilize them into the purification utilizing the affinity chromatography. So, now in today's lecture we are going to discuss about the different aspects related to the generation of the receptor or the ligand, and how you can be able to couple them to the matrix with the help of the different types of the chemistry what is being developed to couple the ligand or the matrix to ligand or the receptor to the matrix, and how you can be able to utilize them for purifications. So, as we can have discussed last time that the receptor or the ligands are actually the making a pair, because the receptor or the ligands are sharing the several interactions. For example, you have the first thing is the receptor is having a 3-dimensional conformation which is exactly complementary to the ligand. So, this ligand is going to fit into this receptor, and then you have the other kind of interactions like the hydrogen bonding, van der Waal or the pi-pi interaction, and in the some cases you may have the salt bridges interactions between the receptor and the ligand. And now, if you want to design the affinity chromatography and would like to purify the protein or the analyte of your interest, you have the two choices. Either you can use the receptor or to the ligand to make the pair with the matrix, which means you can couple the ligand or the receptor to the matrix, and if you use the receptor to be coupled onto the matrix, then you are going to produce the protein along with the ligand. Similarly, if you couple the ligand to the matrix, then you are going to generate the protein to the receptor, okay, so that the receptor and ligand are going to make the interactions. Now, the question comes, under what conditions you are going to prefer to use the ligand or the matrix. The sole purpose of any chromatography technique is to make the purification as easy as possible, as economically viable, which means it is should not be very costly to perform, and the other parameter is that it should not require the extensive infrastructures. So, in many cases whether you use the receptor or the ligand depends on the considering these parameters. So, in few cases you are going to couple to the ligand, or in few cases you are going to couple to the matrix, the receptor to the matrix. Now, let us discuss how you can be able to generate the receptor or the ligand, and how you can couple them to the matrix, so that you can be able to utilize them into the different types of chromatography techniques. For example, whether it is a bioaffinity chromatography or the pseudo affinity chromatography. So, generation of the receptor, although we are saying the generation of the receptor, but it is more or less like the receptor or the ligand, which is very the same thing actually. So, the receptor molecule present onto the matrix can be produced either by the genetic engineering, isolation from the crude extract, or in the case of antibodies, it is produced in the mouse or the rabbit model and purified. So, you have the three options to generate the receptor or the ligands. you have the genetic engineering. So, you can actually genetically clone the gene which is responsible for a particular type of receptor, and that is how you can actually, and then subsequently you can purify that receptor by using the recombinant DNA technology. You can put some tag or some other kind of features, and then you can be able to purify the receptors using the other chromatography techniques. The other option is that if the protein is not, or the gene is very difficult to clone, or it is not been isolated so far, but the protein is available, then you can be able to isolate the protein from the crude extract. That can be done simply by either using the conventional chromatography techniques like ion exchange chromatography or gel filtration chromatography or hydrophobic interaction chromatography, or that can also be done by the antibodies, which means suppose I want to make the insulin receptor or I want to isolate the insulin receptor. So, I have two options, either I will break open the cells, I will prepare the lysate which is actually going to contain the insulin receptor, and then I will go with the fractional fractionation of that particular lysate with the help of the different types of chromatography techniques, ion exchange chromatography, hydrophobic interaction chromatography, gel filtration chromatography, and then ultimately I am going to get the pure insulin receptor. The second option is that I can just use the antibodies which is directed against the insulin receptor, and then I can just take out the insulin receptor from the crude mixture, and that is how I can get the receptor, and that can be used for subsequently to for the other affinity chromatography techniques. For example, I can use the insulin receptor to simply purify the insulin from the blood. The third is that where the places where you are actually going to use the antibodies. So, antibody is actually making a pair with antigen. So, you can be able to generate the antibodies which is maybe against any receptor or ligand, or so it can be generated against the antigen. See, the recombinant DNA technology, the genetic engineering and the isolation is already what we have discussed, or the genetic engineering is what we have discussed in other courses. So, we are going to discuss about the generation of the antibody. Now, the generation of antibody is a multi-step process where you are, first thing what you require is the antigen. So, antigen is a proteinaceous substance which is actually be present, which is the external agents, or it is something which is going to cause the immune response into the particular animal or the animal. So, antigen could be immunogenic or the non-immunogenic. So, the antigens which are non-immunogenic are called as the hapten. So, in those cases where you are working with the antigen which are coming into the category of hapten, for example, the drug molecules. So, if you are trying to generate the antibodies against the for example, the aspirin. So, if you are trying to generate the antibody against the aspirin or the chloroquine or artemisinin or any other paracetamol, in those cases the antibody is not going to be developed, or these molecules are non-immunogenic, or they are not going to cause any immune response into the animal. So, because of that they are fall under the category of the hapten. So, in those cases what you are going to do is you are actually going to convert a hapten into a immunogen simply by coupling that to a protein, and because of that the hapten is also going to be convert into a immunogenic molecule, and then that complex can be used as antigen, and that complex can be prepared or can be used to immunize the animals. Irrespective of whether you are working with the hapten which is being converted by coupling it to a protein to antigen, or you have the natural antigens which or the protein of very high molecular weight, then process these antigens so that they will be good enough to immunize the animals. So, we will discuss that what are the different procedures or the processes you have to do when you want to process the antigens, so that it will be ready for the immunizations, and then you are going to immunize the animals with the help with these antigenic preparations. And once the animal is going to be immunized, it is going to be immunized 2 times. One is you are going to put the primary injections, and then you are going to give the booster injections, and that actually is going to create or develop the antibodies into the animal. Once antibodies are being developed, you can be able to collect the blood of the animal, and then from the blood you can be able to collect the serum, and then from the serum you can be able to purify the antigen or purify the antibodies. At this stage where you are actually going to see the antibodies in the blood, you can be able to detect these antibodies, because then only you will be deciding whether I should give the additional booster dose or whether I should just go ahead with this particular level of antibodies in the blood and start collecting the blood. So, that is the whole scheme through which you can be able to generate the antibodies in the animal. So, let us summarize this. First you have to do is you have to prepare the antigen. So, whether the antigen is the hapten or the proper antigen, which is like bigger proteins. If it is a hapten, you have to couple that to the protein, so that it will become the protein like protein drug complex, and that is also going to be processed for the antigenic preparations, so that it will be ready to be given for the injections into the animal. And once you are going to immunize the animals, you are going to immunize the animal 2 times. One is the primary injections and then the secondary injections. With the help of the primary and the secondary injections, you are going to generate the antibodies. Then you are going to test the antibodies with the help of the ELISA, and once you see that the antibodies are being developed with the help of the ELISA, then you are going to collect the large quantity of the blood from the animal, and subsequently you are going to collect the serum from the blood, and that serum is going to contain the antibodies. So, you can be able to purify your antibodies or the antibody which you have developed against this antigen, and that you can use subsequently into the affinity chromatography. So, let us discuss these procedure in detail. So, the first is the preparation of the antigen. So, the antigen required for the development of polyclonal antibody is approximately 2 milligram. It is required for the multiple injection to induce the robust immune response, and the preparation of antigen has the following steps. The first is that you have to generate or produce the antigens. So, you have the 2 options. One, you can use the recombinant DNA technology, which means you can actually be able to clone the antigens, and that is how you can be able to produce the antigen in large quantities and purify. The second option is that you can be able to isolate the antigens. So, there are 2 different approaches to isolate the antigen from the E. coli overexpressing cells. For example, you can purify the antigen under the native conditions. So, even if the antigen is being produced by the recombinant DNA technology, the isolation of the antigen is having the 2 options. One is that the purification of the antigen under the native conditions, where what you are going to do is you are going to overexpress the antigen into the E. coli expressing cells, and then you can be able to perform the different types of column chromatography to purify the antigens. So, under these conditions the antigen is going to be functionally active, and that can be used for immunizations. The second is that the isolation of antigen under the denaturating conditions, which means, so there are that what happen is in some cases the antigen what you are overexpressing, the E. coli is not soluble, which means it is not present in the supernatant, and because of that this antigen cannot be purified utilizing the conventional chromatography techniques. So, in those cases you are going to isolate the antigen under the denaturating conditions. The second is sometimes the antigen is very produced in a very small quantities. So, if you use the conventional chromatography, you are actually going to lose the protein at every step. For example, I think we have discussed that in the previous lecture, that even if you use a conventional chromatography, you might lose more than 50% protein. So, if your protein production in the E. coli cells is very low and you are using the conventional chromatography techniques, the overall yield is going to be so low that you will not be able to get the enough quantity of the antigen to immunize the animals. So, under any of these conditions, you can be able to purify the antigen under the denaturating conditions, because even if you have a protein which is present in the native conditions or the denaturating conditions, the overall immune response does not vary, because even if you have a protein under the native conditions, it is going to be denatured while you are going to process it for the injections. So, it hardly matters. So, in the isolation of antigen under the denaturating conditions, what you are going to do is you are going to do the electroelutions of the antigen from the SDS page. So, let us see how you can be able to isolate the antigens from under the denaturating conditions using the electroelutions. So, isolation of antigens from the SDS has the multiple steps. The first is that you are going to get the antigens overexpressing cells. Then you are going to prepare the lysate, and then this lysate has to be resolved onto the SDS page. So, in this case you are not going to use a mini SDS gel. What you are going to use is a very large SDS page, so that you will be able to load somewhere around 2 to 5 ml of lysate. Normally in a typical SDS page, what you load is somewhere around 10 to 50 microliter, whereas in this case we are going to load 2 to 5 ml of lysate. So, it is going to be a big gel, and so you have to run a maxi-prep gel. So, this is going to be on a maxi-prep gel, and then what you have to do is you have to identify the region of your interest, which means if you run the SDS page, you are going to get different types of bands, and then you have to identify the region where your antigen of interest is present, and then what you have to do is you have to cut this region and bring out the protein band. So, then you have to cut the protein bands. Once you got the protein band which is present in the gel slice, you will be able to do the electroelutions, and in the electroelution what you are going to do is you are going to take this gel block, put it into a dialysis bag, and then you put it into a beaker, okay, and in the beaker you are going to add, or you are going to perform the electrophoresis. So, you are going to put the positive and negative electrode across this beaker. So, what will happen is that because you are going to do the electrophoresis, the protein band is going to start migrating from the gel block. So, you can imagine that even if I have a protein block into the gel block, and if I put the electrophoresis, which means I am going to put the negative on top and positive onto the bottom, this protein band which is negatively charged because it has the bound SDS is start going to migrating, and after some time the protein is going to come out into the solution. So, it will migrate and then eventually is going to fall into the outside buffer, and then what you do is you just collect the outside buffer, and that outside buffer is going to have the proteins, whereas the gel slice is going to be present inside the dialysis bag, and then from here you can be able to just concentrate and collect the antigens. So, this is very simple procedure, and this is very easy to perform, and it is so it is very routinely being used to isolate the antigens which are very difficult to overexpress or which are not soluble, or other kind of conditions what we have discussed. So, to explain you this procedure more in details, I would like to take you to my laboratory, and then I will show you a demo where we are going to perform these steps, and so that it will become more easy for you to follow these steps in your laboratory, and you will be able to isolate the antigens under the denaturating conditions. In Today we are going to give you a demo about the electroillusion. So electroillusion is a technique through which you can be able to isolate the antigen from SDS page. So you can see this is a typical SDS page through which we are interested to isolate this particular band. In ideal situations, you are supposed to not stain these gels, but since we are just showing it for the demo purposes, we are going to show you with this particular gel. So before you start the experiment for the electroillusions, what you require is you require a SDS page where you might have resolved your sample or the protein. You require the blade so that you will be able to cut the your protein of your interest. Then you require a dialysis membrane, you require a beaker full of water, and then you also require the rubber bands so that you can be able to tie up the dialysis membrane. So let's start the this electroillusion experiments. So in the step 1, what we have to do is we have to first see that what is the band what you have to isolate. So for that purpose, what you have to do is you have to first check the protein, the site of the protein or the antigen where the site is present. So for example, in this we have run the purified protein, and this is the major band what we have to isolate. So what you have to do is you have to cut this particular region of that SDS page. So what you can do is simply go with the lane. For example, if I want to run isolate this particular protein band, so what I'll do is I'll just first cut the SDS page from the side like this, and then I can just cut from the top and the bottom, and then you can be able to remove this particular gel block, and that should be good enough for eluting this particular protein or this particular antigen. If you are interested to isolate the protein for a very large concentrations, then in that case what you can do is you can simply merge all these wells and you can run a very large quantity of the protein. So once you have cut the block and now your block is ready for the electroillusions, then in the second step what you have to do is you have to prepare your dialysis membrane. So this is a typical dialysis membrane what we use in the laboratories. So what you see is it is actually a plastic kind of thing. So it is not a plastic, but it is look like as, so it has two folds actually, or the two slips actually, but they are stick to each other. So before you start, what you have to do is you have to just cut the dialysis membrane, and what you can see is that it is actually stick to each other. So both the ends are stick to each other. So to get this dialysis membrane to be in a working conditions, what you have to do is you have to put this dialysis membrane into a beaker, and then you fill this beaker with a water. Okay, and then you just put this into a microwave so that it will get warm up. So while this dialysis membrane is going to be warm up, what happens is that the two sheets of this particular dialysis bag is actually going to be removed from each other, because when the water is going to boil, it is actually going to remove the surfaces what is being used to adhere these two membranes, and as a result it is actually going to give you a bag in which you can be able to place your dialysis, your SDS, the band that we have just cut from the SDS page, and then we can be able to do the electroillusions. So now what you see is that I have boiled the dialysis membrane, and now dialysis membrane is both the layer of the dialysis membrane is being removed. Now what you can see is that the bag is ready for putting the the membrane or this SDS block. So what you can do is you can simply open this and then you place this block inside the membrane, inside this bag very carefully, so that you should not damage the protein. And once this is being put inside, then what you can do is you can just put the rubber band from both the ends. So you can just put the rubber band on this side, and you have to be very careful that it should not be leaky or should not cause any problem. So when you put the rubber band on one side, and then you put the rubber band on the other side, and in between what you can do is you can just simply fill a small amount of the buffer into this, so that your protein will be in the water instead of, and by doing so you can also check that there is no leakage actually. And because if there will be a leakage, the protein is going to come out from here instead of going via the electroelutions. So you can just add 1 or 2 ml of your buffer, and that should be good enough. And then you can do is, what you can do, you can close it from the top as well with the next rubber. So if you are want, you can even use the you can use the dialysis clips as well, so that but if you can use even the thread as well. So the purpose is only you can just close this, so that you can make a bag like this. Now you can see that the band is inside, okay. And what I'll do is, now what you have to do is you have to just submerge this into a horizontal gel apparatus, okay. Fill it with the buffer, and it doesn't matter what buffer you use, but it should be conductive so that the current should flow. And then what you can do is you can just close this and connect it to the cathode and anode. So you connect to the black and the red, okay. And now you just run it on 100 volt. So you can just run it on 80 to 100 volt, and what you see is now that the electrophoresis is going on. And when the electrophoresis will be going on, what will happen is that this protein band is actually going to start traveling into this particular SDS page. So because we have cut the SDS page at this end, the protein band will run into this direction. But after this, actually there is no gel actually. So what happen is the protein is actually going to fall outside the gel, and because outside we have already covered it with the dialysis membrane, so it will actually fall into that particular liquid what is present into the dialysis membrane. And now your protein or the antigen is going to be present into that particular solutions. So now we are going to continue with this electroelution or this electrophoresis for at least for another 1 or 2 hours, and then we are going to see whether there will be electroelutions or not. And then after that, what we are going to do, we are going to remove the rubber bands. We are going to open the dialysis membrane from one end. We are going to collect the supernatant, and then you can just concentrate that supernatant, and it is actually going to give you the protein of your interest. So this is all the few steps what you have to do if you want to do the electroelutions to isolate the antigen in a large quantity under the denaturating conditions from SDS page for the production of antibodies. So I hope you might have understood all the steps, and it could be helpful for you to advance your work. With this demo, I hope you might have learned how to electroelute the protein bands from the gel slice, and you will be able to utilize this demo to extend your work or to perform the additional experiments related to this. Now let us move on to the next aspect. So once you have prepared the or produced the antigen in the large quantity, then you have to prepare the antigens for the injections. So the preparation of the antigen for the injections, what you are going to do is you are going to combine the 100 microliter of antigen, where you are going to have the 100 to 150 micrograms of protein with an equal amount of fluids complete adjuvant to a final volume of 200 microliter. What you have to do is you have to take the 100 microliter of the antigen and 100 microliter of the fluids adjuvant, and you have to take the Fruent's complete adjuvants, then it is actually going to be 200 microliter, and these 200 microliter you have to mix thoroughly to obtain a emulsion using a syringe or the pipette. So, once you mix them, the Fruent's adjuvant actually contains the detergent, and it actually contains the oil, and it also contains the Mycobacterium tuberculosis cell wall. So, Fruent's adjuvant is actually going to stimulate the immune response, and it is actually going to activate the system, so that the system is ready to take up the antigen. But since you are making the emulsions where the oil and the water is going to mix together, and because of that the antigen is going to be trapped into the small vesicles. So, what happen is, when you take the oil and you mix it with the water, it is actually going to make the emulsions where your antigen is going to be trapped within this emulsion. So, because of that the antigen is going to be released very slowly from the site of action, and that is the purpose for which you are actually producing this emulsion, so that the antigen should be keep immunizing your animals for a very long time. Because what happen is, if the antigen is going to be cleared from the site of your injection very soon, then you are not going to challenge the animal for a very long time. So, that antigenic response will not go to the very large chunk of the B cell or T cell. It was going to spread to very small population of the B cell and T cell, and because of that the overall immune response is going to be very less. And why we are adding the Mycobacterium tuberculosis cell wall? Because the Mycobacterium tuberculosis cell wall is very immunogenic. So, when you inject the primary injections where you have the Fruent's adjuvant, and which actually contains the Mycobacterium tuberculosis cell wall, the cell wall is actually going to activate the system, so that the large amount of the immune machinery is going to be present at the site of injections. After 4 weeks of your first injection, which means the primary injections, you inject the first booster dose. So, in the booster dose, what you are going to do is, you are going to again prepare the antigen in the same way, except that this time you are going to use the incomplete adjuvant instead of the complete adjuvant. So, in the first round you are going to use the 100 microliter of your antigen and 100 microliter of Fruent's complete adjuvant, whereas when you are going to do the booster injections, you are going to use the 100 microliter of antigens and 100 microliter of the Fruent's incomplete adjuvants. What is mean by the Fruent's incomplete adjuvant is that the Fruent's incomplete adjuvant does not contains the Mycobacterium tuberculosis, because now after 4 weeks the machinery is already been under activated state, the site of your antigen action injections is already having the lot of cellular machineries. So, now what you do is, you replace the antigen, the Mycobacterium antigen, and you inject your own antigens, against which the immune response is now going to be developed. So, as a result, in the secondary injections the antibodies are going to be developed against your own antigen. Now, repeat these booster injections 4 to 5 times after every 4 weeks to generate a robust immune response and a development of the memory B cells, which are actually going to generate the antibodies. Now, what you have to do is, before you do the injections, you have to you do some procedures. So, in vivo immunization of rabbits, before immunizations, take out 5 to 10 ml of the rabbit blood from the rabbit before first injections, and that is called as the pre-bleed, which means you are going to take out the blood from the mouse or the rabbit, and that is called as the pre-bleed. Incubate the sample at 4 degree at 30 minutes and allow the blood to clot. Centrifuge the sample at 7000 G for 10 minutes, and you can be able to collect the serum and store it at -20, and this serum what you are going to collect before immunization is called as the pre-immune serum. The pre-immune serum is actually going to tell you the level of antibodies which are already been present in the rabbit, which means before the immunization, which means the antibodies which are being present in the rabbit against other antigens, not the antigen of your interest. After this, the whatever the injections you have prepared, either in the complete adjuvant or the incomplete adjuvant into the rabbit, which is the NZ strain, which is the New Zealand strain. So, and during this step, either use a helper to hold the rabbit or use a restraint device to hold the rabbit. So, you have to you have to hold the rabbits very strongly, so that you will be able to inject the emulsions, either the primary injections or the secondary injections. So, you have the two choices, either you ask someone to help you and hold the rabbits, or you can use the restraint devices where you have to just keep the rabbits, and then rabbit will not be able to move, and then you will be able to inject the antigen on the back side of the rabbit in the form of the button, which means if this is the rabbit, you have to inject somewhere on the back side of the rabbit, because this is the site where you have enough space and enough area, which so that it will be not be a problem. And then you have to inject it in the form of button, which means you have to inject at one site, make it a button like this, and then you go to some other site, you inject, then you go to other site. So, all these are called as buttons actually, and that actually is required because the antigen is now going to release from these buttons on a very slow mode, and as a result it is actually going to cause the robust immune response and the large production of antibodies. Now, once your primary injection is over, then you can do a booster injections. The procedure is almost the same, except that you are going to use the incomplete adjuvant versus the complete adjuvants. Once you are done with the booster injections, then you can determine the antibody titer. What is mean by titer is the amount or the level of antibodies against the antigen of your interest. So, in that case, you take out the 5 to 10 ml of rabbit blood from the ear vein of the rabbit, incubate the samples at 4 degree, and then you collect the serum, and you determine the antibody by a indirect ELISA. So, indirect ELISA anyway we are going to discuss in a later module when we are going to take up the techniques related to the immunology. So, that time we are going to discuss about the indirect ELISA, and we are also going to show you how to perform the indirect ELISA as well. After that, once you are sure that the antibody titer is very high, you can be able to collect the blood and prepare, and you can prepare the serum. So, for that you take out 20 to 30 ml of the rabbit blood from the ear vein, or the large quantity of blood can be drawn after the cardiac puncture. So, you can have the two choices, either you draw the blood from the ear vein, which is actually not going to be a terminal procedure, but if you are looking for a very large quantity and you are sure that the experiment is now been over and you do not need any more antibodies, then you can be able to collect the blood from the cardiac puncture, which means you can just make a hole into the heart, and that is how you can be able to collect very large quantity of the blood from the rabbit, but that is a terminal procedure. After that, the rabbit is not going to survive and he is going to die. Irrespective of the locations, then you have to incubate the sample at 4 degree for 30 minutes and allow the blood to clot. They centrifuge the sample at 7000 G for 10 minutes, and then you can be able to collect, because once you collect the blood and you allow them to clot. So, once the blood is going to be go for the clotting reactions, you are going to get two fractions. One fraction where you are going to get the blood cells, which is actually going to form the clot, and the second place where you are going to get the liquid part, which means that is the serum. So, that is a yellow color serum you are going to get after the clotting reactions. Now, once you have produced the antibodies into the rabbit, and as we have discussed all the theoretical aspect related to this particular technique, I thought of showing you how to produce the antibodies in a when you go to the animals, or when you go to a animal house and try to do yourself. So, for this purpose I went to the Central Drug Research Institute, and then I took the help of the institute, because we do not have the animal house. So, that is how we have taken the help of the other institutes, and I went there, and then I have while they, because they were doing this immunization and the antibody production steps. So, I got the chance, and then I actually made a demo video, so that where the scientists are actually explaining each and every step what we have discussed so just now, and that actually is going to helpful for you to understand how the antibody is being produced inside the lab, inside the animal house, and how you can be able to immunize the animals. And I hope that the demo video what is going to be helpful for you to understand the practical aspect of the antibody generation as well.
I am Amog Ransasdubite. I work in CSIR, CDR, I don't know. And in today's demo, we will be discussing different steps involved in generation of antibodies in rabbits. So, for the first step, we require several things like France complete adjuvant, here it is from Sigma. We need a microemulsifying needle which has two openings connected with a fine needle. We need antigen which is purified and filtered, so there are no contaminations. It is a sterile solution of antigen. Then we take out some of the France complete adjuvant in Nippendorf and then mix them together. Since this adjuvant is oil based, it doesn't mix easily with the watery system like the antigen is in PBS. So therefore we mix them rigorously and forcefully. For that purpose, we take these two, we mix this emulsion and we mix this PBS and PBS containing antigen and the adjuvant, oil-based adjuvant. After mixing them, we take out in a needle. Using a needle, we take out in a syringe like this, and then we fix the microemulsifying needles into it. Attach another syringe into it. Like this. So once you have filled your antigen and the adjuvant in this needle, you push it here, and then you keep pushing from one side, keep pulling from another side, keep pushing from one side and keep pulling from another side. So this process forcefully pushes your material, I mean the oil and the antigen, through this fine needle, and with that, in that process, the emulsion is formed. Emulsion can be called as water in oil or oil in water, because both are in the same concentration, same volumes, so you can call them anyway. So the emulsion, by this method the emulsion is formed. So for ready reference, we have already prepared the emulsion. This emulsion looks like white. Initially it was two phase, and then slowly it has turned into single phase. Now you can push this emulsion from one needle to another side, and from another this syringe to another syringe. So this process creates very good emulsion, which doesn't separate out later on when you are ready to inject. So how do we check them? So for checking purposes, we drop one of this emulsion, drop a drop of emulsion on the water surface like this. If emulsion is not formed perfectly, this will spread out. Otherwise, it will not spread. So this is a check that your emulsion is formed correctly. So once you find that this drop is not spreading, your emulsion is actually ready for injection. So this was the process by which we prepare the emulsion for injection. So this is the first step of preparing the emulsion. So now let us understand why we prepare the emulsion. You have checked that emulsion is formed. Now the purpose of making the emulsion, because you have antigen and antigen, through antigen you can raise antibodies, but after emulsifying them you actually make the antigen releases slowly. So it is a sustained release kind of preparation, so that the antigen is exposed to system in a systematic manner, so that the more and more memory cells can be is generated, and that's the sole purpose of having emulsion. Otherwise, if you inject antigen as such in EBS or in other watery system, it will be spread out in the body and it will be cleared out by the immune system readily, and no memory cells will be generated. So this is the main purpose of preparing the emulsion. So now we have prepared the emulsion. We have come to animal house. This is our rabbit, which will be immunized, and before immunization we have to take pre-immune bleed, so that we can compare later the serum and the anti-serum. So we will now start how we immunize it. So now we are preparing to immunize. The first and important thing is in all these animal processes is we have to avoid the pain to the animal. So for that purpose we have to strain the animal, because we have to inject. So we strain the animal in a way that it has less and less pain, and the movement is also less. So we will inject this into the emulsion into the thigh. So to catch hold of both the legs, then we have to sterilize the area using alcohol. We have to look at the thigh muscles. They should be cleanly visible. Skin should be cleanly visible. There are two kind of injection that we give. One is intradermal and another one is subcutaneous. So today we'll be doing subcutaneous injection. This is our emulsion that we have prepared by microemulsifying needle. We have seen right here. This is the area where we would like to inject. We have to take out all the airs from the syringe and the needle. We have taken out, clean the area again, and then we apply some antiseptic powder. Here it is betadine powder, so that the infection cannot develop later on. We sprinkle some of it at the area of injection. And then slowly release, leave the animal relaxed, and it is immunized. To strain the animal in a towel or this kind of cloth. So the advantage of having this cloth to strain is the animal has its claw outside of this cloth, and then it cannot move. So we have to restrict the movement. Pain is similar in the strainer and in the on this cloth. So we use this image. Now we will strain the animal on this cloth. We will keep the animal relaxed on this cloth and we will strain it. Make sure that the ear are outside and the animal is trained properly so as to reduce its movement, and now it is ready to bleed. We will bleed the animal from this mid ear vein and we rub it so that it gets loosened up and the circulation is faster. The vein will also expand and more flow will be there. So this is the method which is normally we apply. When the vein is properly visible, this is the mid ear vein from which we bleed. We will slightly sterilize it using alcohol and using a 20 gauge needle which is wide enough to give sufficient bleed. We will prick the vein and collect the bleed. Then to stop it, we just with less and less pain, you can collect the bleed like this. Now we have to make sure that no further bleeding occurs. Then we will wipe out whatever bleed is outside. Using sterile water, we wipe out all the blood here and there. If any spillage, we will wipe out all over using water so that the vein becomes cool and gets shrunk. We just check still it is bleeding, so keep it pushed. Until the bleeding stops. So now I think the blood has stopped coming out, and then now we apply some antibiotic. Here in this case it is betadine powder, so that there is no further infections or inflammation in the rabbit, and this also ensures that this is erythema, and if there is any inflammation it will have some pain, so it will avoid that kind of pain also. So we have isolated approximately 10 to 12 ml of blood. This will give us approximately half of the volume of the blood serum. This will be coagulated. The blood will be coagulated at 37 centigrade for 1 hour, and then it will be kept at 40 centigrade for overnight, so that the clot is shrunken properly and the serum is maximally taken out, and then isolate the serum, add some preservative like sodium azide, and keep it at -20 or -80 as per the requirement, and then we also test simultaneously the titer of it and the specificity of it using ELISA test and the Western blot test respectively. Now you can see this whole of the animal, this animal is very relaxed. It has, I think it has very less pain or a negligible pain. And this is actually very important for handling when you handle animal, that in whatever procedure you go through with animal should be ensured not to have pain. Maybe you can, if the procedure is painful, you understand it is a painful procedure, you anesthetize it. Since this procedure is not painful, we have not necessary. So that this is very important step to ensure that the animal has it should be relaxed. So in whole of the process, I think you have got to know all the steps of animal development in rabbit, and we have to be prepared immersion. We injected the immersion. We isolated the blood after giving sufficient booster doses, and whole of the process is in the I think you have understood most of these processes, and you like it.
So, this demo video is probably be very helpful for you to understand the each and every aspect, especially the practical aspect related to the antibody productions. Then let us move on to the next aspect, that is the coupling of the receptor. So, once you produce the receptor either by the recombinant DNA technology or to isolate the antigen the receptor from the crude mixture using the conventional chromatography, or you have produced the antibodies by the from the animals, you have to perform the coupling of these receptors to the matrix. So, in the coupling reactions you have the 3 steps. One, you are going to first activate the matrix, then you are going to use the coupling reactions, you are going to do a coupling reactions with the help of the reactive groups present onto the ligands, and then you have to deactivate the remaining active groups which are present on the matrix, which means first you have to take the matrix, you treat it with the chemical so that it is going to be activated. So, it is going to be activated, which means you are going to develop some functional groups, then you are going to add the proteins or the receptor whatever. So, you going to add the receptors. What will happen? It is actually going to take up all the receptors what you are going to add, and that is going to be coupled. But it actually has more number of functional group compared to the number of receptors what is going to be coupled onto this particular matrix. So, what you have to do is you have to inactivate these functional groups so that it should not take up additional molecules whatever is present in the crude mixture, because if that happens then you are actually going to couple the 2 particular different types of molecules. One is receptor, the other is the non-specific molecule. So, because of that you have to block these sites, or you have to block these groups some way, so that it should not bring the additional protein molecules or additional molecules to facilitate the non-specific binding of the antigens or other kind of molecules, because if that happens the affinity chromatography will not going to be very specific. So, there are multiple ways in which you can be able to couple the receptor to the matrix. The first method is the epichlorohydrin mediated receptor coupling. So, epichlorohydrin activates the polysaccharide matrix by adding the oxirane group with a 3 carbon alcohol group spacer arm. So, what you have is you have the polysaccharide matrix, then you incubate it with the epichlorohydrin, and that actually is going to generate the activated matrix, and this activated matrix is having the oxirane group. Now, what you do is you add the receptor which contains the primary amine, which means the protein which is actually going to contain the NH group. Okay. Now, the receptors are coupled to the matrix by a thioester or the secondary amine linkage, it can be able to couple the hydroxyl group containing receptor molecule as well by a ether linkage. So, you have two options, either you use the receptor which contains the OH, or you can use the receptor which contains the primary amines or the primary amines or the secondary amines, and that actually is going to couple to the matrix, which means you are going to get the coupling of the receptor to the matrix, and then ultimately once your coupling is over, then you can add the large quantity of the glycine, because the glycine is also having the amine groups. So, glycine is going to inactivate all the activated groups which are present onto the matrix, which means all these groups are going to be blocked by the glycine, and that is how you will be able to inactivate all the functional groups, and that is how you will going to avoid the non-specific binding. The second is the carbamide mediated receptor coupling. So, in the carbamide mediated receptor coupling, the carbamide reacts with the matrix containing the carboxyl group to form the isourea ester. The activated matrix is then allowed to react with the receptor molecule containing carboxyl or the free amino group. Receptors are coupled to the matrix by the secondary amine linkage, which means you take the polysaccharide matrix, which means you can take the dextran or saccharide matrix which is actually going to contain the carboxyl group, and then you add the carbamide. The carbamide is going to activate and generate the isourea esters, and then if you have the primary amine, the primary amine is going to react to this carbonyl group, and that is how it is actually going to form the amide linkage, and that is how your receptor is going to be coupled to the matrix, and this is going to be released from, or the dialkyl urea is going to be released from the matrix. Now, after this is done, again here also you are going to incubate the matrix with the large quantity of glycine to inactivate the additional functional groups. Now, let us discuss about the operation of affinity chromatography and how and what are the different steps are present. So, operation of a affinity chromatography, the first step is the equilibration. Affinity chromatography material is packed in a column and equilibrate with a buffer containing high salt, which means around the 0.5 molar NaCl to reduce the non-specific interaction of a protein with the analyte. So, if you remember, compared to all other techniques, the affinity chromatography is the only technique where you are going to have the very high salt concentration, so that you should not you should avoid the interaction of the analytes to the matrix by the non-specific interaction, because the matrix is also made up of sugar. So, it also can provide the functional groups which where the protein, other protein may also can interact by the isoelectric interactions or hydrophobic interactions or even some other kind of non-specific interactions. So, to avoid that, you have to keep very high salt concentration, so that the ionic interactions and the van der Waals interactions or hydrogen bonding, mild hydrogen bonding should not take place. Then what you have to do is you have to do a sample preparations. The sample is prepared in the mobile phase and it should be free of suspended particles to avoid the clogging of the column. The most recommended method to apply the sample is to inject the sample with a syringe. So, the first thing is you are going to generate the affinity column, which means you are going to produce the matrix, then you couple it with the help of the coupling reactions. Then you are going to equilibrate and apply the samples. So, once you apply the samples, your antigen is going to bind to these receptors, whereas the all other antigens which are present in the sample will not going to bind, and once you do the washing steps, they are going to be washed away. So, the third step is the washing. So, in the washing step, you are going to flow the buffer which contains very high salt concentrations, and as a result the non-specific proteins which are or non-specific analytes are not going to bind. So, that is how it is actually going to remove all those proteins, and then you are going to do the elutions. So, in the affinity chromatography there are many ways to elute an analyte from the affinity column. The first is that you increase the concentration of the counter ligand, which means you can be able to increase the antigens in a very high quantity. So, if you do that, the antibody will switch to the antigen instead of binding to your receptor or binding to the your analyte, and as a result the analyte is going to be released. The second is you can change the pH or the polarity of the mobile phase. If you do so, then also you are actually going to disrupt the salt bridge interactions and hydrogen bonding and van der Waals interactions, and that is how it is actually also going to affect the interaction or the affinity of the receptor to the ligands. Then you also can use the detergents or the cheotropic salt to partially denature the receptor to reduce the affinity to the bound ligands. So, the third is that you can use some denaturating conditions. For example, you can use the detergents or the cheotropic salt. Cheotropic salt means you can use the gonadinium hydrochloride, which is actually a denaturating agent. So, if you put the gonadinium hydrochloride, it is actually going to denature the proteinaceous receptor or the ligand, and as a result it is actually going to eventually, because the receptor or the ligand, if they will not be able to maintain the 3-dimensional conformations, they are actually going to lose the affinity for the counterparts, and as a result the analyte which is binding to the matrix with the help of that particular receptor is going to be released from the column. But this third method is not very much recommended, because eventually the if you go with the third method, it is actually going to give you the antigen or the analytes in a denatured state. The fourth is the column regenerations. Once you are done with the chromatography, then you are going to do a column regenerations. So, after the elution of the analytes, the affinity chromatography requires a regeneration step to use for the next time. The column is washed with the 6 molar urea or gonadinium hydrochloride to remove all non-specifically bound protein. The column is then equilibrated with the mobile phase to regenerate the column. The column can be stored at 4 degree in the presence of 20% alcohol, which is containing the 0.05% sodium azide. So, once you are done with the washing step, then you can just put the counter ion or counter ligands, and that actually is going to become petite for the bound ligand, and as a result the ligand is going to be eluted from the column. So far we have discussed about the bioaffinity chromatography, or we have just discussed about how to generate the receptors to prepare a affinity chromatography column, and very briefly we also have discussed about the generic steps, what is present or what is being used to run the affinity chromatography. Now, in the subsequent lecture we are also going to take up the few specific examples from the bioaffinity chromatography or the pseudo affinity chromatography, and then we have also going to discuss how to perform these chromatography techniques and how you can be able to exploit the affinity chromatography to solve the specific experimental questions. So, with this I would like to conclude our lecture here.